Hi all, Tony here from Immortality Cryptocurrency. Today we're at New Atlas. Article reads, New gene editing tool reduces errors by nicking DNA, not cutting. There is always plenty going on in the gene therapy space. What we're looking for is the best vector and the most precise means of gene editing. So it's very important that we stay up to date and harness the power of gene therapy. A cluster of human hematopoietic stem cells that have been edited with the new spacer nick technique. This kind of colony formation could only happen through successful editing, the team states. Curing aging, age reversal, life extension really depends upon gene therapy. Genome stability, a system to go through the genome and correct mutations, a system to go through the body and repair mitochondria, DNA mutations, a system to manipulate the epigenome, specifically expression. As important as the CRISPR-Cas9 gene editing tool has been, it has a relatively high error rate, which can introduce potentially harmful mutations. Researchers in Germany have now developed a more refined tool that reduces errors by nicking DNA instead of cutting it. There's all kinds of derivative CRISPR versions. Each of them have their pros and cons, but there's probably now we're getting to about maybe 15 different derivatives. Many of them use the CRISPR-Cas9, some of them use other methods. Cas12, J, SA-Cas9, ST-Cas9, NM-Cas9, FN-Cas9, CJ-Cas9, SC-Cas9, Cas-X, Cas-Y, Cas12a, CPF1, Cas1214a, high-fidelity Cas9, ESP-Cas9, SP-Cas9, HF1, hyper-Cas9, FOKL fused, DCAS9, XCAS9, DCAS9, Cas13a, Cas13b, Cas13d, Superfly. And there's also Cas-Clover, zinc finger talon, other methods, base editors, and prime editors. There's also the vectors and the vector technology, which is key to delivering the payload to the cell. The hope is to repair mutations, jumping genes, other mutations, and even possibly to the stage where oncogenes are identified and repaired. The challenge is, imagine how safe the therapy has to become in order for it to be used trivially in vivo or for it to be used in vivo safely. But it's so essential. Unlocking this ability, gene therapy. Currently, there are 6,000 known genetic disorders in humans, although it's likely that the number is much greater. So really, a watershed technology for the possibility of treating 6,000 known genetic disorders and possibly a whole lot more. So it is super essential that we obtain some capacity to perform gene therapy in vivo. CRISPR is one of the most groundbreaking inventions of the century, with the potential to revolutionize gene therapy for a range of diseases, as well as improving the yield or nutrition of crops, creating beneficial microbes and a variety of other uses. It works like a pair of molecular scissors, cutting out problematic genes and slotting in more helpful ones. Now, the use of gene editing for crops and plants is not advised, and we do not support the use of genetically modified crops and seeds. The problem is sometimes it can target the wrong section of DNA and make the change there, known as an off-target mutation, which can potentially trigger health issues. Even if it does get the right target, the DNA repair process can go wrong and lead to what's called an on-target mutation. Preventing both of these issues was the focus of a new study by researchers at the Max Delbruck Center of Molecular Medicine and Humboldt University of Berlin. The team modified these molecular scissors to be a bit more gentle, making a fundamentally different type of cut. Rather than making one cut that slices through the entire double-strand DNA. The new tool makes 2 smaller nicks, each cutting 1 DNA strand. A built-in spacer keeps those nicks a safe distance apart between 20 and 350 base pairs. Our experiments with hematopoietic stem cells and T cells have shown that this is the optimal distance for minimizing both on-target and off-target mutations, said Dr. Van Trung Chu, co-last author of the study. Any shorter and we risk cutting through the entire DNA molecule despite the use of 2 separate scissors. A general comparison of classic CRISPR-Cas9 and the new spacer-Nick gene editing techniques. So he claims on-target mutations to be less than 2% using this method versus greater than 40% with CRISPR-Cas9. They claim the off-target effect is minimal or even absent. Now, in our field, we don't want to change DNA to make somebody's brown eyes blue or somebody's skin color darker. We just want to fix the mutations. In tests on cells in lab dishes, the team found that the new spacer-Nick tool was about as effective at making edits as conventional CRISPR. Between 20 and 50% of treated cells were repaired, but importantly, the new tool was much better at reducing errors, with on-target mutations occurring in under 2% of edits with spacer-Nick, compared to over 40% for CRISPR-Cas9. Off-target mutations, meanwhile, appear to be a rare, if not non-existent, occurrence in our approach, said Chu. The team hopes that future work will test the spacer-Nick editing tool in animals before moving on to human trials. One of the first potential targets is to treat inherited blood disorders. The research was published in the journal Science Advances. Science Advances says precise CRISPR-Cas9 mediated gene repair with minimal off-target and unintended on-target mutations in human hematopoietic stem cells. While CRISPR-Cas9 is a key for the development of gene therapy, its potential off-target mutations are still a major concern. Here we establish a spacer-Nick gene correction approach that combines the Cas9 nickase with a pair of PAM-out sgRNAs at a distance of 200 to 350 BP in combination with adeno-associated virus, serotype 6 template delivery. Our approach led to efficient HDR in human hematopoietic stem and progenitor cells, HSPCs, including long-term HSCs and T cells with minimal NHEJ-mediated on-target mutations. Using spacer-Nick, we developed an approach to repair disease-causing mutations occurring in HBB, ELANE, IL-7R, and PRF1 genes. We achieved gene correction efficiencies of 20 to 50% with minimal NHEJ-mediated on-target mutations on the basis of an in-depth on-target assessment. Frequent unintended genetic alterations induced by classical CRISPR-Cas9 were significantly reduced or absent in the HSPCs treated with spacer-Nick. Thus, the spacer-Nick gene correction approach provides an improved safety and suitability for gene therapy. So this is another potential way, a potential area of investigation to investigate whether we can use a system to correct mutations and keep the mutation rate at a young level rather than changing anything, possibly 70% less mutations throughout the organism, which should result in better function. We'll write this in our gene therapy section and take note of it. So, which gene therapy platform is the one? Is it CRISPR, zinc finger, talons, or another one? And which vector? Let's hope we see some in vivo results soon. Immortality Cryptocurrency, over and out.